This is Mir Shaukat Hussain, teacher from Zone Bekepora, District Badgam, UT Jammu and Kashmir. The topic of today's lesson is from the chapter Heredity and Genetics. The topic of today's lesson is Mendel's contribution to genetics, which shortly we can call as Mendelism. As we know, G.H. Mendel, Gregor Johann Mendel, has contributed a lot towards genetics and heredity, because of which he has been regarded as father of genetics. Today, I have selected this very topic as I think this will be very helpful for the students of secondary as well as higher secondary standard. The objectives that will be fulfilled in today's video are like this. First, we'll see what is genetics, that is, what is the definition of genetics. After that, we will have contribution of Mendel to genetics, that is Mendelism. And finally, we will see what were the conclusions made by Mendel, which became the law of genetics. Let's begin with what is genetics, that is, what is the definition or meaning of genetics? Genetics is the branch of science in general and biology in particular, which deals with the study of heredity, variations, and inheritance. Heredity means transfer of characters or transmission of characters from one generation to another or from parents to offsprings. Variations are the differences among the individuals, and inheritance means the ability of the offspring to receive and express characters or genes or genetic material from parents. So we can say genetics is the branch of biology which deals with the study of how characters are transmitted from one generation to another generation, how characters get expressed in offsprings, how are variations created among individuals. Now, after knowing what is genetics or what is the meaning or definition of genetics, we'll now study the second one, that is Mendelism, or what has Mendel contributed to genetics. Mendel carried experiments for 9 years on garden pea with scientific name Pisum sativum to work out the rules of genetics. Mendel, because of his tireless work of 9 years, came with some conclusions which later on became laws or rules of genetics. Because of his contribution towards genetics, he has been regarded as father of genetics. For the sake of simplicity or easiness, we will study Mendel's contribution or Mendel's heading under different headings. The first will be selection of experimental plant. Selection of experimental plant. In this heading, we'll study for experimentation which plant Mendel selected and why. Second heading will be selection of contrasting characters. In this heading, we'll study what are contrasting characters and what contrasting characters were selected by Mendel for his experimentation. Third heading, experimental technique. Experimental technique adopted by Mendel. What experimental technique is adopted by Mendel to carry out his experimentation? And fourth heading, that is crosses done by Mendel. What are various crosses done by Mendel? Fifth and last heading of Mendelism will be conclusions made by Mendel. Selection of experimental plant. In this, we'll study which plant Mendel selected for carrying out his breeding experiments and why he selected the very plant. The experimental plant that Mendel selected is garden pea, that is Pisum sativum. He selected this very plant for experimentation because Pisum sativum or garden pea possesses some features. The first feature is pea is an annual crop, so results will be quick. The second reason of selecting this very plant as experiment plant is pea can be maintained and grown under controlled conditions. Third, pea plants have number of contrasting characters. Fourth, the flowers of pea plants are bisexual, that is hermaphrodite, possess both male as well as female sex parts. And fifth and last but not the least reason of selecting the garden pea or Pisum sativum as a plant for his experiments is the flowers of garden pea are large enough that they can be handled easily. Because of these reasons, Mendel selected garden pea or Pisum sativum as his experimental plant. Selection of contrasting characters. We'll see which contrasting characters were selected by Mendel for carrying his experiments. But before that, we should know what are contrasting characters. Contrasting characters are those characters which are opposing to each other or which exist in two opposing tendencies. As an example, height is a character. It has got two contrasts or contrasting characters. Tall and dwarf are short. So tall and dwarf or short are two contrasting characters. Likewise, another example is seed shape. Round and wrinkled. These are two contrasting characters of seed shape. Mendel, for his experiments, selected seven pairs of contrasting characters. That is, he selected 14 contrasting characters. Those very 14 contrasting characters are mentioned in the table. Let me read them out. First is stem length, or you can say height of plant. It has got two contrasts, tall and dwarf. Second is pod shape. It has got two contrasts, inflated and constricted. Third is seed shape. It has two contrasts, round and wrinkled. Next is seed color. Again, two contrasts, yellow and green. Next is flower position. It has two contrasts, axial and terminal. Flower color. It has two contrasts, purple and white. Lastly, pod color. It is green and yellow. These seven pairs, or you can say 14 contrasting characters, were selected by Mendel for carrying his breeding, or you can say breeding experiments on Pisum sativum. Now let us see what experimental technique was adopted by Mendel for carrying his various experiments on Pisum sativum. Mendel used a three-step technique in his experiments with Pisum sativum. The three steps of his experimental technique are selection of pure parents, production of first generation by cross pollination, and third step is production of second and subsequent generation by self pollination. As we know that Mendel has selected 14 contrasting characters. So Mendel bought 14 varieties of pea, 14 varieties of pea plants with 14 contrasting characters. But to satisfy himself that all the 14 varieties of pea plants are true to their character, he allowed all the 14 varieties of pea plants to self-pollinate for many generations and eliminate any offspring that was not true to its character. Pure pea plants thus obtained were selected as parents for cross breeding. After satisfying himself that the 14 varieties of pea plants are true to their character, Mendel started cross pollinating the pea plants differing in contrasting characters to obtain the first generation. In this setup, Mendel cross pollinated two pure pea plants differing in contrasting characters After cross-pollination, the seeds were obtained, and the seeds, when sown, germinated into new generation of pea plants. This generation was called as first filial generation, that is F1 generation. Next step is production of second and subsequent generations by self-pollination. After producing first generation, that is first filial generation of F or F1 generation by cross-pollination, Mendel produced second generation or F2 generation by self-pollinating F1 generation, and third generation, that is F3 generation, by self-pollinating F2 generation, and F4 generation by self-pollinating F3 generations, and so on. So Mendel produced F1 generation by cross-pollination, and rest of the generations he produced by self-pollination. Now we'll see what were the various crosses done by Mendel. Mendel performed a number of crosses, but we will study two types of crosses. Firstly, monohybrid cross, and secondly, dihybrid cross. Now, what is monohybrid cross? Monohybrid cross is the cross in which we study the inheritance of only one character. So the cross in which we study inheritance of only one character is called as monohybrid cross. In one of the monohybrid crosses, Mendel cross-pollinated a pure tall pea plant with a pure dwarf pea plant. The F1 generation obtained was tall, that is, all the pea plants in F1 generation were tall. When F1 generation was self-pollinated to obtain F2 generation, in F2 generation, some pea plants were tall and some were dwarf. But the tall and dwarf pea plants were in the ratio of 3:1. This ratio, that is 3:1, is known as monohybrid ratio. Now, what is dihybrid cross? Dihybrid cross is the cross in which we study the inheritance of two characters only. That is the cross in which we concentrate only on two characters is known as dihybrid cross. In one of the dihybrid crosses, Mendel cross-pollinated a pure pea plant with round and yellow seeds with a pure pea plant with wrinkled and green seeds. The pea plants in F1 generation were all having round and yellow seeds. After allowing F1 generation to undergo self-pollination, the pea plants in F2 generation were of four varieties, that's were of four kinds. First, pea plants having yellow and round seeds. Second, pea plants with having round and green seeds. Third, pea plants with wrinkled and yellow seeds. And lastly, pea plants with wrinkled and green seeds. All the four varieties of or types of pea plants in F2 generation were in the ratio of 9:3:3:1. This ratio 9:3:3:1 is regarded as dihybrid ratio or dihybrid cross ratio. After conducting a number of crosses, Mendel made some conclusions which came to be known as rules or laws of genetics or rules or laws of inheritance. Mendel from crosses made four conclusions which came to be known as laws or rules of inheritance. First is law of paired units. Second, law of segregation. Third, law of dominance. Fourth and last law, that is law of independent assortment. Let's first take The first law, that is law of paired units. This law states that every character of an individual is controlled by two genes called as allelic genes. Out of two allelic genes controlling a character, one gene is paternal, that's from papa, and the other gene is maternal, that's from mama. So this means that every character of an individual is controlled by two genes. One gene for every character comes from papa, and the other gene controlling the character is derived or comes from mama. The allelic gene controlling dominant character is denoted by capital letter, while as it is recessive character is denoted by the same but small letter. For example, allelic genes controlling tallness or allelic genes in case of pure plant controlling tallness will be capital T, while in case of dwarf pea plant they will be small t, small t. Second law, that is law of segregation. Segregation simply means separation. As in previous law, we came to know that every character of an individual is controlled by two genes called as allelic genes. This law states that during gametogenesis, that is gamete formation, the allelic genes controlling a character segregate or separate, and each gamete receives one gene out of each allelic pair. Coming to third law of inheritance or rule of inheritance, namely law of dominance. This law states that during cross pollination, F1 generation receives genes for each character from both the parents. But out of each allelic pair, only one gene finds its morphological expression by dominating the other gene. The gene which finds the morphological expression or which gets expressed in F1 generation is called as dominant gene, and the gene which does not find its morphological expression or is not expressed in F1 generation is called a recessive gene. So this law states that genes can be dominant or recessive. In presence of dominant gene, recessive gene can't express itself. Out of the four laws or rules, the first three laws, that is law of paired units, law of segregation, and law of dominance, were derived by Mendel from monohybrid crosses. But the last and the fourth law, that is law of independent assortment, was derived by Mendel from dihybrid cross. This law, that's law of independent assortment, states that during a dihybrid or polyhybrid cross, the allelic genes of each character are distributed independently to the gametes. That is, the allelic genes controlling various characters segregate independently and move to the gametes independently. This was all from this video lecture. I hope you would have understood things clearly. We will meet inshallah shortly in next video. Till then, God bless you and take care.